from the Danish Research Center for Magnetic Resonance, and he will speak about post-mortem and clinical diffusion imaging, and we're really happy to have him here. Tim.
Thank you. So, this is working. So I will start going down here, because then I can point on the screen. That's much more easier for me. Otherwise, we have no interaction on my fantastic slides you will see here in 2 seconds. So I've been invited to talk about post-mortem and preclinical diffusion imaging here. So one of the questions diffusion MRI, we need some kind how to validate our stuff from the in vivo. And it could be very nice to do it in post-mortem. So the question is, why is this not working? Make it work, please. So the thing is here that now it works. Sorry. Yeah. So is post-mortem bring a reliable model of in vivo? That is one of the important questions that we are going to raise. Today we can use these diffusion MRI techniques that allow us to map in the brain network with tractography. We can map the DTI, the microstructure features, and we can also, that we heard earlier today, using biophysical modelings that allow us to go more into details of the microstructure in mapping axon diameters. But it's very challenging to do it in vivo in the human brain. And if we could go to post-mortem, we also have a direct access to histological features that allow us to validate these estimates that we try to derive from diffusion MRI. That can be electronic microscopy, showing the axons, the myelin, and we can have myelin information from myelin stains, and we can also use invasive tracers techniques here to visualize tractography. For the last part here, that is non-invasive techniques. So therefore, we often use our small friends, the animals here, in a preclinical setup. So what is like the good thing to go to post-mortem compared to in vivo? Yeah, but first of all, we do not have any motion. We have no pulse artifacts. Scanning time can be unlimited, so we can have high signal to noise, and we can have high resolution, more or less at the same time. We can use alternative sequences for diffusion-weighted imaging. Today, in vivo, we use the fast EPI sequences that are challenged with distortions and lack for signal to noise, but we can use very slow ones here that are because we have a lot of time. And also another thing is that we can use much more powerful MR scanners. And here what we are thinking about is especially the hardware, the gradient strengths allow us to use in these scanners that are not really available today in the clinical settings. We have, of course, some challenges. One of these are tissue degeneration. We have influences of the fixative. The fixative is used to keep the tissue stable after death. And also we have a terminology here, what I introduce later on, and that is about short-term instabilities. It's an effect that we apply to handling the tissue, and then we start scanning. Then we can introduce these short-term instabilities in our data set, and we do not want to have that. When we start to set up an ex vivo imaging pipeline, it could look like these 4 steps. In the first, up at the top, we have a living human subject. Then we have the fixation state, the first one, where we use some type of fixative. We use a fixation procedure. We have in stage 2 storage. So the storage is the time where we have the fixated brain storage during the scanning sessions. And then we have the preparation step, where the preparation is what we prepare the tissue before scanning. And that we need to decide on the fluid, the resting time, and the environmental temperature need to be adjusted when we prepare it for scanning. Then we have the last stage here is actually when we acquire the data set of the scannings. And here we need to choose a sequence. The parameters need to be adapted. And also we have scanning temperature to handle. And the final end, we'll have a high quality diffusion-weighted imaging data set that is much more nicer, crispy than we can see in vivo. So let's go through the different stages here in this pipeline. The first one is the fixation. So why is it important to fixate? Well, that's because we want to avoid tissue degeneration. So after death, the brain tissue and cells then begin a self-destruction process due to their own autogenous enzymes. And this process is called autolysis. And how it looks like is this is an example. So here we have an example of electronic microscopy on a tissue sample where our fixation process failed. So that means that our tissue was not fixated at time that we expected, but we collected the EM anyway. Here we have a situation of tissue that was correctly performed, fixated, and we can see the EM. And we can see huge differences between these two environments. We can actually see the cell membranes here, but we can see also the myelin layers are loosened around it. And if we have a situation here, due to this degeneration of the tissue, then we have an environment that is not like in vivo, and we cannot use our models in this type of ex vivo as a model of in vivo. We want it to be something similar to this one. There are different ways of fixate a brain. We can have immersion fixation and we can have perfusion fixation. The immersion fixation we can use on human and animals. Simply we take out the brain after death, put it into a can of fixative, and let it passively diffuse inside this tissue. It will take about, dependent on the size of the brain, it will take about 3 to 4 months to have a full fixated tissue. We can also do it more efficiently with perfusion fixation. So in perfusion fixation, we can only do it on animals. What we have is that we have the animal very deeply anesthetized, then we open in and have a tube into the heart, and then we flush through the whole vascular system. First, we remove all the blood with a PPS, and then it follows by the fixative. When we have been doing this, we take out the brain, and then we post-fixate the tissue. In that way, we ensure that the tissue is fixated almost before the tissue actually discovers that it's dead. So we have different types of fixatives. We have one we call the class of aldehydes. There's the formaldehyde and glutaraldehyde. Normally we are using it in 4% and 10% solutions. So these fixatives are like cross-linkers. And for example, the formaldehyde is a very small molecule that has benefits in being a very fast penetration into the tissue and cross-link the tissue to ensure it to be stabilized over time. The glutaraldehyde is a slower one, but you can, depend on what kind of staining you want to do on the fixated tissue, you can combine the features of these two. The penetration about formaldehyde is about 1 millimeter per hour. And then we also always, when we validate microstructural imaging, we talk about shrinkage. When you do fixation, it has been reported that there's only a minor shrinkage appearing from in vivo to the fixated brain. It's about 5%. So the big shrinkage we observe when we are talking about validation in microstructure, for example, axon diameters and other kind of stuff, is coming when we do the processing of the tissue for histology, where we use a lot of different chemicals that make the different tissue compartments to shrink. So how fast does the brain actually start to degenerate? So we have this, what we define as a post-mortem interval. It's a time from the death to when the fixation starts. And it has been shown that there's minimal autolytic changes in EM after 6 hours. And after 24 hours, you start to see changes. And it's very dependent on the temperature of the tissue after death. So this is an example after 6 hours EM, where you can actually see the myelin start to be loosened here around the axons, but it's very small effects. The big effects are coming about 24 hours or 48 hours after. So how does it actually impact our diffusion measurements? So I did a small experiment. We have a monkey brain here. We draw a small region of interest in the corpus callosum in splenum and calculated the mean FA within this one. In vivo, we have a FA of about 0.8, but in the perfusion fixated of the same brain, where we have like the post-mortem interval is 0, we have a bit higher FA. If we now take, this is another brain, but we do the same kind of analysis, then we can see that if you use immersion fixation, then it takes time for the tissue to be fixated, and you can see in the same region that you have, or you can risk to have a lowered FA value, indicating that the microstructural features are not as nice as in vivo. Just to play a comment why the ex vivo is higher, it's simply because we have a higher image resolution here compared to in vivo, so we have a minimal risk of part-to-volume effects to, for example, the surrounding CSF. How the post-mortem interval impact diffusion can also be shown in this study by Helen Jacquel, who show that in a rat brain at time 0 after death, and then repeated scannings for 1 up to 14 days. And we can see, for example, the first eigenvector, you can see it, the lambda, or the diffusivity, actually decreases significantly after time, pretty fast. An indication also here show that it significant impact tractography. This is an example where they're seated in corpus callosum, and we get a nice result across corpus callosum immediately after death, so we at this time point. And after 14 days, the tissue has totally degenerated, and you cannot really trust your tracking results. So this is an indication how important it is to ensure the quality of the tissue to minimize the post-mortem interval. So we can say for ex vivo tissue to be a good model for in vivo, the post-mortem interval must be as close as possible to 0. Now we have a fixated brain, and then we need to store it. So the question is, how stable is the tissue over long times? And we investigated it for about 3 years. We followed 3 different brains with repeating scannings. And between the scan sessions, we placed the tissue in about 5 degrees. We can see here for the different brains, this is the mean diffusivity, and where black is white matter and red is gray matter. We can see that the variation across time is small and is less than the variation between brains. And we have the same indication here in the principal direction. We can see that the diffusivity is constant over time, indicating that the tissue has stabilized. We saw before that if you have autolytic effects going on during time, then the principal direction, the diffusivity will decrease significantly. So now we have the stories, and now we need to prepare the brain for MR scannings. And in this sense, we have some tips and tricks you can use. First of all, when we do diffusion, one of the things we are spending a lot of time on is to ensure have a high signal to noise. And one of the tricks here is always to wash out the free fixatives from the tissue. So it's not the surrounding fixative, it's a fixative that's inside the tissue. And the reason to do that is actually shown, demonstrated in this nice study, where this is done in cortex. So there was unfixed tissue where you have relaxation of T1. And you can see if you have different type of formaldehyde, you can see the T1 when you have fixated here did not really change when you have washed out the fixative from the brain. But in T2, which is important when we do diffusion MRI, that is directly linked to our signal to noise, we can see in the unfixed tissue we have about 0.1, 10 milliseconds. But here we have with the fixed, when we are not washed out the fixative in the tissue, then we have a significant decrease in the T2. However, if we just take the sample of the sample and just put it in PBS for about a week and wash out the fixative, then we actually can restore T2. So this has been shown for cortex, but exactly the same things yields for white matter. So always place take your tissue, place it in a can with PBS about a week before the scanning time. Then there are tricks if you want to do acquisition very fast. Then you can actually take your fixated brain and soak it in gadolinium. And what Helen Agrell has shown here, that what you do is with this soaking of the gadolinium, with time you have lowered you can see here that the T1 lowers, but the T2 appears to be stable over time. What we also can see here. So that means that with a shortened T1, then we also can shorten the TR, and then we can speed up the acquisitions. This, of course, you need to have very powerful gradients, but often we have that on the preclinical systems. But if you push a system with a fast acquisition, you also heat up the gradient, and because the tissue is just placed inside the coil, you need to ensure that the temperature is constant during this scanning when you push the scanner. It's good for tractography, and it's also been shown that have minimal impact on DTI measurements. but you need to be aware of the gadolinium is a big molecule, so it's placed in an extracellular space. So it's not an effect that you introduce in both the intercellular and extracellular space, and that might impact your microstructure imaging when you try to find out estimates of the signal contribution from the intercellular and extracellular spaces. How it impacts, I'm not really sure, but you need to be aware of there's a chance that it can impact your measurements. Now we have the tissue ready, then we need to make the brain placed in the RF coil so we can start the scanning. But one of the questions is then we need to have some fluid around the brain, and it will be a very bad idea to have a big can of fluid and then the brain swimming around here. Then you have introducing a lot of motion artifacts. So we need to avoid that. We need to ensure a very stable mechanical setup when we acquire our diffusion data set. And here I will show my way to do that. So I need some Lego. I bought it myself. And then we have some tape and the sticky stack here. We take the brain. This is a monkey brain. We put PBS inside in the brain in the plastic bag, and just ensure there was no air bubbles around it, and try to squeeze out almost all the water from the brain. This is how it looks like. Then I construct a mechanical setup that fits into the diameter of the coil when also the brain are placed on top of it. Then I take my brain, put it upside down here, and then I take this microporous tape. So this is very important. When you use this type tape here, it must not have any electrical features. It should be tape that will not apply any forces onto the brain when you have put it around it. Because if you have it, then you will squeeze and you will introduce distortions to your microstructural measurements. So put it around it very gently and ensure that it's just stabilized the brain on top of this one. Now it looks funny because I put it upside down, but actually there's an idea there because we have gravity. So water inside the brain will be thrown down. So in that way, I will ensure not to introduce any air bubbles during the scan times, and we are doing long scan times. So if I put it the upside down, then all the water will be placed between the two frontal lobes, and I will have introduced over scan time a lot of air bubbles, and that is not good. Now the last trick here, you take the sticky stacks here, put it on the corners on the Lego, put it into the RF coil and press it, and then you have a very nice mechanical setup for ex vivo imaging. Now we have our setup. We need to verify that it's reliable. And one of the things is when we handle the brain is a bit soft. So when you handle it, it's like introducing some mechanical forces, and it takes time for the tissue to stabilize. And we are measuring things at the micrometer length scale. So we need to verify that our setup does not include these short-term instabilities due to this handling. And how you can do it? You first acquire all even slices with different directions. I use 60 directions, and then after a time lag, then I collect all the odd slices. Then when I concatenate these slices, then I have a time lag of 22 hours between even and odd slices. This is an example of when I place the animal into the scanner. This is time 0, and we can see if I do this, then I have these stripes between the slices. This indicates that we have some mechanical effects that changes the signal, the diffusion signal. If I wait some time scanning, then I can see that these stripes start to disappear. And after 15 hours, I have almost homogeneous between even and odd slices, as I would expect it to be. So actually, this is the time point where I can start to acquire my diffusion-weighted imaging dataset in high quality without these short-term instabilities included. One could then say it's temperature. But you can actually place a small ROI here in the water. This is it. And you can see already after about 3 hours, the temperature effects here has stabilized inside the scanner. So it is mechanical effects. So this here, 15 hours, sounds as a long time. With the mechanical setup I showed you before, it takes about 6 hours for these short-term instabilities to disappear in our setup. But check your setup to see you do not introduce this into your measurement. Now we have the scan. We have everything ready now, and we put it into the scanner, and we need to optimize our MR sequences to ex vivo imaging. First of all, it has been shown that the anisotropy Ex vivo is similar to in vivo. I also showed you with in the corpus callosum. But the diffusivity is decreased about 60 to 80% in white matter and gray matter. That means that we need to adjust the p-value to handle this lower diffusivity. So for normal DTI in ex vivo, where we need to acquire a general p-value, it needs to be about 4,000 seconds per square millimeters, compared to about 1,000 in humans. And if we do not do that, then we are really challenged. So this is an example where we look at the fiber directions here in the same scanning session, where I have collected a hardy data set of a p-value of 1,000, 4,000, and 15,000. And you can say about 1,000, we can see that the directionality is not really clear. It's not homogeneous spatial, make much sense. But when I do it to 4,000, then we start to see a clear pattern. Crossing fibers get consistent in the tissue. And then you can start to increase the p-value, and you just get a more sharper profile of your fiber reconstructions. So this also clearly demonstrates that you need to have a high p-value ex vivo to have something that is similar in vivo. So you also need to remember we have higher p-values in the preclinical scanner, and that pushes the gradient system. Therefore, you also, again, need to take care of the temperature control in your scanner, because you just have the brain tissue placed in the middle of the magnet. Now, this is how we see it today. This is like a typical clinical DTI, and this is like a post-mortem. Of course, it takes longer time to acquire it, but what is actually the possibilities with this ex vivo imaging compared to in vivo? But before we start to look at that, then it's important to also know that we have some challenges here. For example, if we have a T1-weighted image here in infrared in a human in vivo, and then we have also an infrared in a post-mortem. This is a monkey brain. We know in vivo we have a gray matter is gray and white matter is white, but in T1-weighted of a fixated brain, these intensities have shifted. So now the gray matter is light and the white matter is dark. This intensity shift does not happen in T2-weighted. So this is an example where we have the B0s, which is a T2-weighted. There, the gray matter is light and the white matter is dark, as expected. And we can clearly see it here on our very low quality of the human data set. But I can tell you this is true. That means, because we have this shift here in the intensities of gray matter, white matter, you cannot do what has been suggested by Van Essen group doing this T1 to 2 ratio imaging, which is an indicator of myelination. That will not work in vivo because of this shift. I put here the diffusion-weighted images. This is a B1000 and corresponding B4000 in a monkey brain. And you can see how the quality here is much higher than we are used to see in a human clinical scanner, where we use the EPI. And this is a slow sequence, a spin-echo sequence with a single readout. It takes ages to acquire, but we have time here to do it. So with this diffusion data set, we can start to look into anatomical features that not always are visible in vivo. And one of these is, for example, the different organization of cortical layers. So we have shown that this is in a pig brain, we have like two rims, an inner rim and an outer rim. The inner rim has low FA values, and the outer rim has a higher anisotropy. We have directionality toward the orthogonal of the cortex, and the lower rim here with the low FA values, looking with DTI, mainly correspond of crossing fibers when you add a fiber model there. And this you can see all over the brain in any kind of animals. And actually has also been shown here by Lloyd, shown in a visual cortex of a human and a piece of it put into a preclinical scanner. And actually they managed to visualize even more layers in cortex based on the profile of the diffusion directionality. So the information is there, it's just to investigate it. It's very interesting. What we also can use this kind of setup with the ex vivo, we can use it for validation. So we can do this, what we call direct validation. So direct validation is one of the most powerful ones because we inject the tracer in the brain, and it's the same brain that we acquire the diffusion-weighted imaging data set onto it and apply the spectrography. So we cannot have any, so there is not really these differences, anatomical differences that we could expect between animals. So this is an example where we have a manganese tracer. So a manganese tracer is detectable with a T1-weighted image. We also simultaneously injected a BDA tracer just for control that we visualized after the post-mortem imaging. And this is an example of the post-mortem where we defined the seed region giving these tracers. Now we have the neuronal tracer. This is the manganese. And we can now compare it quantitatively with the spectrography here. We can overlap them and see how they correspond. And we can see if the projection signs are right or wrong. And from this kind of experiments, validating spectrography allow us to give us an insight into the challenges that we heard previously about the spectrography, which is often challenged in regions from crossing fibers, but also it's challenged in how to terminate in different cortical regions. Of course, this is validation, and you can visualize this for different spectrography methods. You can look at this Knut et al. But we also comment that you can see here with this tracer here also have its limitations. So we call it a gold standard, but actually it is a kind of silver standard because you can see we injected here in this region, but manganese has a tendency to diffuse in the extracellular space and make the tracks more broad. So it's very hard to have high sensitivity using this kind of experiment. You can also do spectrography in your human postmortem, and this has Carla Miller from Oxford shown. She has been put up to an experiment where at Christian that allow her to acquire such high quality of the human brain and show that she can extract major fiber bundles from the brain. But as I told you before, these postmortem brains have postmortem interval which is pretty large, and her analysis also show that we introduce different FA values that are dependent on the post-mortem time, so the time after death. For example, in corpus callosum, we can say from 12 subjects that we have a large variation in FA values, much larger than what you would expect to have in vivo. This indicates that this tissue actually is not the same shape or looks like what would we expect in vivo. But we can turn it around. So instead of imaging the human brain, then we can actually apply microstructure features or this kind of acquisitions in the living human brain and then use the ex vivo to verify this. And this we have been doing in the microstructural imaging where we verified the ActiveX that Gary told about before the lunch break. We show it's possible to extract axon diameters indices in a human brain using a clinical scanner. We have low, high, low axon diameters across corpus callosum that also is described by histology. And we could, just to verify this experiment, we can have an experiment with a much longer scan time, and we can do the same acquisition in postmortem. And here you can see that the microstructural features or the measurements estimates here are more clearly seen on the monkey brain. The reason is here that we are using a preclinical system that allows us to have a stronger gradient strings, and that show up to be more important to get a robust estimate of axon diameters. So we investigated this, the impact of gradient strings on these microstructural features, and because we have ex vivo, then we can do this kind of very long scan times. And when I'm saying very long scan times, it's about 1 week or more. To verify, to use this protocol optimized with different gradient strings from 60, 140, 200, and 300, and to investigate how this impact on our microstructural model to estimate axon diameters. And we can see about 200 to 300, then we get robust estimates of axon diameters across corpus callosum. But if the Gmax gets too low, then we cannot really fit it and we cannot trust the results. So this is a possibility that we could do ex vivo. But I also have to say that we use the same, almost the same biophysical model ex vivo as in vivo, but we also observe changes. First of all, when we design acquisitions for ex vivo, we have to adjust it for this lower diffusivity that we observe in ex vivo compared to in vivo. Another thing is when we investigated this, our original in vivo model did not really fit to the signal profiles that we obtained in the ex vivo. And it showed out that this could be explained by a small fraction of the water molecules that are like trapped during diffusion times. And that small compartment we do not see in vivo. So that is added to this ex vivo. From where this small compartment are coming from, it's not really known. But one could imagine that when we're going to fixated tissue, then we stop all the physiological properties of cellular exchange in different cells. So that could be the glial cells and all this. And maybe this change in permeability of cell membranes can introduce this additional compartment that we observe. It could also be that when you use formaldehyde, it has been shown that it introduced small bleeps here on the cells. So this is a culture, but it has been shown also to tissue. But the question is this really a source that can explain this compartment we observe? I do not know. So the last point here I want to show the possibilities what we also can use ex vivo for. So we have these clinical scanners which appears to low resolution, but on some scanners you have the ability that you can actually interpolate the data, and also in different software packages allow you to interpolate your data to higher image resolution. And in that sense, the tissue looks much nicer. It actually seems that we can extract anatomical features not seen on an original image resolution. And is that really true that we want to estimate with, prove with ex vivo? So what we did here is that we took on one brain, scanned it in one long scan session. This actually took 2 weeks from image resolution of 1 millimeter, squared 0.6, 0.4, and 0.25. And then we can use this low image resolution, interpolate with different methods, and compare it, validate it with the actual high resolution data sets. The most clear effect is seen here. You can actually start to see there's small cortical regions here where we can see this lower rim of low FA values. When you interpolate here, we use a 7-order interpolation, then you start to see that these appear much more clearly in the images. And that corresponds to what you actually can see with the higher resolution original data sets. You also see other smaller details. So the details, for example, we have tracks here, which are part of volume. But when you go into interpolation, you start to see small rims of low FA values. Low FA values integrate the interface between gray matter, two fiber tracks with different directions. And that one you actually also can see at the higher resolutions. What you also see here is like an effect here. You have general blurring effects when you do interpolation. So if the image resolution is too low to extract the anatomical features, then you also will introduce blurring. So you can take this data set, so this is an original human data set, and interpolate the raw data set. It's not interpolating the FA maps, it's interpolating the raw data set and then reconstruct the DTI. And then here you can see in the clinical, you see small changes here in the interface between tracks. The shapes of the hippocampus also looks nicer. But the small changes you observe. So yes, it does matter to do interpolation. But why is it that it works? It's because that we have a spatial dependency of the anisotropy from the voxel to voxels due to the brain connections that connect the different brain regions. So therefore, and it's this directionality that actually introduces this additional information. So therefore, you do not gain any effect of just interpolating the raw FA map because there's no directional information. The interpolation method used has impact, and you should go to the higher order one if you want to gain something from this one. But there's also a lot of interpolation artifacts you can observe. We have ringing artifacts due to the flexibility of the interpolation method. You can observe hotspots where the interpolation method tries to reconstruct something from noisy data set. You can have failed DTI areas, and you also include this blurring I showed you. So therefore, if the scanner suggests you to interpolate your data, then do not do it. Always save your original data, and then you can do the post-processing, whatever you want to do afterward. So in conclusion, ex vivo diffusion MRI of the brain tissue is challenging, but if you have insight into the key element of tissue degeneration and onto the ex vivo setup, it opens a unique translational setup for translated door into anatomical details that we have not seen with in vivo. The first thing is that you need to ensure the tissue quality so it's as close as possible to in vivo. Minimize post-mortem interval. Use perfusion fixation, and immersion fixation is not optimal. Acquisition, wash out the fixative to increase the signal to noise, ensure mechanical stability, and ensure the data quality. The validation purposes here, you can adjust the scanner parameters to ex vivo due to lower diffusivity, and then you can compare it with invasive tracer spectrography. You can test advanced microstructure features and introduce and verify new MRI technologies. And then finally, many of the data sets I have shown you, you can actually download them and try them out. We publish them free on our homepage. And thanks to my group and all the collaborators and the funding.
Thank you very much. So we have time for some questions as we started a little late. So who wants to ask a question? Yes, please.
So you are always interpolating the raw DY data, each independently or jointly together? The different independently. So you one gradient, you interpolate it, and then the other gradient. Okay, that's it. Thank you.
Okay, Siavash?
Very interesting talk. I would like just to get this clear. So you're saying that when we do perfusion, for example, perfusion fixation on human brains, then it doesn't matter whether we start immediately, let's say 10 minutes after death, or 24 hours whenever, we will always have the problem that because of the diffusion process, we won't get, we will see always this blurring of these myelin sheets and thus a reduction in FA value. Is that what you're saying?
Yeah. First of all, we cannot do perfusion. That's when you do it on animals. So it's immersion fixation. So you take the tissue, put in.
I meant diffusion.
Yeah. So it's true. Yeah. So
diffusion.
It's because we only have from the surface, and then it need to go into the brain, and that takes time. Yeah. And that time, the tissue still degenerates.
And does it? So is there a difference whether you start then immediately or a few hours after death, or
of course,
hours?
It's important to do it as close to post-mortem time as possible. Mm-hmm. And when you get the brain out, keep it cool because that slows down the degeneration process, mm-hmm. and put it in a fixative.
Do you have a feeling for basically what is more important? The effect, what happens after the basically after it is in the fixative, or the effect which happens between death and the time you put it into the fixative? So
I'm concerned of the situation where the tissue degenerates. So if you choose immersion fixation, it takes time for the tissue to be fixated. That I'm concerned. Of course, I want to have it perfectly at time 0 and fixate the tissue, because what I would like to use it for is for validation of techniques. Yes. So that's the main idea. So keep the post-mortem interval to 0.
Thank you very much. Very interesting.
Yes, please.
Have you had the opportunity to look at to scan brains immediately after death, but in situ before fixation? So you have 6-hour window or so. So just immediately after death, scan them, and to avoid the issues of fixation.
Yeah, that you can do, and then you do not have the fixation process. You just need to be check your because the brain start to maybe you get I don't know what's happening in the brain, but you have a small time slot there where you can actually measure the tissue, which is more close to in vivo than when you do the fixation. So this is a possibility.
Just to add to this, I want to advertise a little project which happens in Brazil, in Sao Paulo, that they set up a 7 Tesla scanner just for this issue. So if you're interested in this, Edson is quite keen in collaboration on this.
So you mean acting as a brain or the data set?
No, not as a donor, but as to set up projects together with them and to do like a sort of validation of tractography.
Yeah, cool.
Any more questions? So thank you very much. And then I'm happy to introduce my co-chair, Anton Beer, who will do the next talk on comparison of diffusion MR imaging and applications to brain plasticity. He's from the University of Regensburg in Germany. And we're looking forward to your talk.